Welcome to our biology lessons with Dr. Hanaa. Today we will solve with each other June 2025, Variant 1, Paper 6. The first question said, a student investigated the effect of concentration on diffusion. We said before that when we find the word of effect of, we know that after this word we can find independent variable. But here they didn't say us the concentration of what. So now we have to search about concentration of what to consider the independent variable. The student used the pieces of agar which contain universal indicator. Universal indicator is green in substances with a neutral pH and red with the substances with an acidic pH. The student placed the pieces of agar into different concentration of hydrochloric acid. And now we reach to the concentration of hydrochloric acid. So this is independent variable. And measured the time taken for the acid to diffuse to the center of each potato of agar. So how can we measure the rate of diffusion? So when we say that this dependent variable, you have to know this dependent variable should be measured. So we have to measure the time taken, and this is dependent variable. At the start, the investigation, the agar was green. This means that this is a neutral. The student used this method. Label for test tube 0.4, 0.6, 0.8, and 1. The table shows the values of 1 mole per decimeter cube of hydrochloric acid solution and distilled water that used to make it a different concentration, of hydrochloric acid. So we have 4 test tubes, one of them this 0.4, 0.6, 0.8, and 1. This is concentration of hydrochloric acid. And they put the volume of 1 mole per decimeter cube of hydrochloric acid solution per cubic centimeter, 4, and then added 6 of distilled water. So the total volume here equals 10. And by adding 6 of hydrochloric acid, they add 4 a cubic centimeter of distilled water. So also the total volume of solution is 10. And when we put 8, they just add 2 cubic centimeter of water to make the total volume 10. And the last one, they add 10 hydrochloric acid with 0 distilled water. Also, the total volume is 10. So we observe here the most concentrated one, which is 1, as they just adding hydrochloric acid and there is no distilled water. Step number 2. Use 10 cubic centimeter syringe to put the volume of 1 mole per decimeter cube of hydrochloric acid shown in the table into test tubes 0.4, 0.6, 0.8, and 1. Use 10 cubic centimeter syringe to put the volume of distilled water as shown in the table into the test tube 0.4, 0.6, 0.8, and 1. And they give us all the volume of hydrochloric acid and the volume of distilled water to make different concentration of acid. A block of agar which measured approximately 1 centimeter times 2 centimeter times 2 centimeter is provided. We have here a problem. When they said that approximately, so it's a source of error because they didn't say us this is exact value. Use the knife. So we have here hazard to cut the agar block into 4 pieces. Step number 5, place all test tubes into warm water bath. He didn't say us this is maintained, and he didn't say us the value of temperature of the water bath and never maintained. So it's a kind of a error. How can we improve it? We have to use thermostatically controlled water bath. Put 1 agar pieces into each of the test tubes and immediately start the stop clock. Observe the color. So we have here other kind of error. Why? Because anything this judged by eye, this is subjective. So how can we improve it? We have to use colorimeter or color chart. So observe the color of agar pieces as they change from green to red. The student recorded the time taken for each piece of agar to become completely red, and we said that this is the dependent variable, and the stop clock from the step 6 are shown in the figure. So the first one, which is 0.4, first concentration, 9 minutes and 37 seconds, 0.6, 7 minutes with a 56 seconds, 0.8 concentration, 6 minutes and 27 seconds, and the highly concentrated one, 4 minutes with 12 seconds. Prepare a table to record the result of the investigation. Convert the times on the stop clock to seconds and record this time in the table. How can we convert it? In front of us, 9 minutes with 37 seconds. So we have to multiply 9 multiply 60 because 1 minute equals 60 seconds, okay, plus 37. Second one, 7 multiply 60 plus 56. Third one, 6 multiply 60 plus 27. 4 multiply 60 plus 12. And finally, the result, we have to put it in the table. The first one equals 577. The second one equals 476. And third one, 387. And the last one, 252. So now we have two different data, so you have to use it to make the table. So we have the concentration of hydrochloric acid solution mole per decimeter cube, and the time taken for the pieces of agar to become completely red by seconds. So you have to put this data in the table by this way. Don't forget to put the unit in the heading of the table, not in the cell. Next question, state a conclusion for the result. When they ask about the conclusion, we have to return back to the objective of the experiment. So you have to know the effective concentration of hydrochloric acid on the rate of diffusion. So when we say that, as the concentration increases, the time taken for each piece of agar to become completely red decreases. It is not a conclusion. It's an observation or description for the table. So how can we convert it to be a conclusion? So you have to say, we have to talk about the rate of diffusion. As the concentration of hydrochloric acid, so the higher the concentration of acid, the increases the rate of diffusion. As the concentration of hydrochloric acid increases, the rate of diffusion increases. This is the conclusion for this experiment. Next question, state the independent and dependent variable in this investigation. We said it and we make it easily to be catched from the experiment. So the independent variable, the concentration of hydrochloric acid, and the dependent one, the time taken for the agar cube to change its color. Next question suggests two improvements to the method used on this investigation. We said that, so we have to keep the temperature constant by using thermostatically controlled water bath. And we have another improvement. They said that approximately, and we said that it's a kind of error. How can we improve it? So you have to cut the agar equal size pieces. We have another one. They didn't repeat this experiment, so they have repeated at least 3 times and taken average. And the last one, so you have to use colorimeter or color chart to help determine the endpoint, because the change in color, this is subjective. This is something that judged by eye. Next question, identify 2 hazards in this investigation. So we said this, the knife is sharp and may cut the hand, and also the using of hydrochloric acid, which is irritant. So we have these 2 hazards in this investigation. Next question, plan an investigation to determine the effect of temperature on osmosis in potato plant tissue. So firstly, the effect of temperature. So this is independent variable that we have to change it on the osmosis in potato plant tissue. So in osmosis, how can we know this? So we just bring the potato plant and cut it into similar size, and we have to measure the initial and the final change in mass, and we can measure the change in mass or percentage change. Now, how can we start this investigation with each other? So we have to prepare and cut 3 potato cylinders using cork powder and also the knife to have the same size and the same length. But this potato should be with the same age and same type. This is also this controlled variable. After that, we have to bring 3 beakers and put in each 50 cubic centimeter. For example, you can add 100 cubic centimeter or any certain volume should be exactly the same. And I put it distilled water. Some student asked to put the concentrated solution, so it's okay of the same concentration because we just have to change temperature. So you can choose the solution that you want, but with the same concentration. I choose distilled water, and maybe other one that want to use sugary solution or salty solution of the same concentration. Then we have to bring this potato cylinders and measure its mass using sensitive balance to get a precise result, okay? And this mass called initial mass, because at the end, we have to measure the final mass and calculate the change or calculate the percentage change in mass. After that, we have to put each cylinder in each beaker and put each beaker in thermostatically controlled water bath to keep the temperature at different values. For example, 10 degrees Celsius, 20 degrees Celsius, and 30 degrees Celsius, okay? And then we have to leave the beakers for about the 60 minutes, okay? If you like to put it in a 30 minutes, no problem. But we have to keep them at the same period of time. After that, we have to get this cylinder from the beaker and use tissue paper to just dry it carefully without squeezing to get exact increase in mass. Finally, measure the mass of potato cylinder using digital balance again, which is final mass. After that, we have to calculate the change in mass. So this is final minus initial, or the percentage change by the rule final minus initial divided initial multiply 100. It is not necessary to write it. So if you wrote it, so no problem. If you don't, it just points that you have to measure or calculate the change in mass or the percentage change. Then we have to repeat it 3 times for each temperature and take an average. The last one, which is safety precaution, you have to wear gloves, goggles, and use tongs because we use thermostatically controlled water bath, and cut on a cutting board to not cut it your hand. Next question. A student used DCP-IP solution to compare the concentration of vitamin C in 5 solutions. Concentration of vitamin C in 5 solutions. The student used this method, put 1 cubic centimeter of 1% DCP-IP solution into a test tube. Use syringe to add drops of 125 milligram per decimeter cube of vitamin C to the test tube. Keep adding drops of vitamin C solution until DCP-IP solution becomes colorless. We know that the original color of DCP-IP is blue, and in the presence of vitamin C, the color change into colorless, or we can say this decolorization of the blue color of DCP-IP. Record the total volume of 125 milligram decimeter cube of vitamin C added. This method was repeated with 4 other concentration of vitamin C solution and state the expected color of DCP-IP solution before. So before starting the experiment, so the original color of DCP-IP is blue. So if we make an analysis for this investigation, so when we put 1 cubic centimeter of 1%, so we have this is controlled variable of DCP-IP solution. With a 1%, this means that this is the concentration of DCP-IP, which is controlled variable. So when they ask about the variable that were kept constant, so we have the volume of DCP-IP, which is 1 cubic centimeter, and the concentration of DCP-IP, which is 1%. The result of the investigation shown in the table, the concentration of vitamin C per milligram per decimeter cube, and the total volume of vitamin C solution added per cubic centimeter. We said that the first column of this investigation, this independent variable, this is the concentration of vitamin C, and we have to put it on the x-axis. And the second column, the total volume of vitamin C solution added per cubic centimeter, and this is dependent variable, which we have to put it on the y-axis. After that, we have to learn how can we draw a line graph on the grid, on the data. So when we draw any graph, everything that on the first column, in the heading of the first column, which is independent variable, this is the label of x-axis, which is concentration of vitamin C per milligram per decimeter cube. So we have to write it as it is, including the measuring unit. And on the second column, the heading is dependent variable. We have to put it on the y-axis as it is with its measuring unit. So the total volume of vitamin C solution added per cubic centimeter. So we have here 4 marks. We have to make axis label. So if you did it in a correct way with their measuring units, so you take the first mark. The second one with a scale. So you have to know what is the least value and the highest value in each. Okay, to make it the correct scale. You have to occupy 2/3 of the graph paper minimum. Okay, so I divided the x-axis with 200, 400, 600, 800, and 1000, and the y-axis with 0, 0.5, 1, 1.5, and 2. This, if we make it, so we took the second mark. The third mark for plotting, when we plot the points, so you have to plot it by using x. So in front of us, 2 options, maybe x, maybe dot with a circle, but which one of them is better? If you have just only one data, you have to use X. But what about if we have two different data? We have to put it on the same graph. So you have to make a key beside it, okay? And use both X and dot with a circle. So if you plot it in a correct way, so you'll take the third mark. The last mark for making a line, draw a line. So we can find this is most of these points so closer to each other, so we can make it line of best fit that passes in between most of them. So now you will take 4 marks. The student tested the sample of orange juice. They found that 1.2 cubic centimeter of orange juice was needed to react with 1 cubic centimeter of 1% DCP-IP. Use the graph to estimate the vitamin C concentration of the orange juice. Show the graph how you obtained your estimate. Please concentrate about this point because from you showing that, how can we get the data? You will take the mark, not just write it. So you have to show them how can we get it. So we will go to 1.2 cubic centimeter. This is 1.2. And make it a straight line using ruler. And just make intersection with the line that you draw. And then drop it on the x-axis to get the value. This is about the 420. So the concentration of vitamin C here, 420. Next question, the student wanted to investigate the effect of storage on vitamin C concentration in apple juice. 3 samples of fresh juice, fresh apple juice, and 3 samples of stored apple juice. So we have fresh and we have stored one. Were tested. So the results are shown in the table. So we have 3 different samples, 1, 2, and 3. So the concentration of vitamin C in fresh juice by milligram per decimeter cube. This is for the fresh one, and the other one which for the stored one. So state the conclusion for the data. How can we know the data? How can we know the conclusion for this one? So if you concentrate, we can find this. All the data, the concentration of stored one less than the fresh one. So storing of fruits that decreases the content of vitamin C in apple juice. The student decided that the result of one sample shown was anomalous. So what's meant by anomalous result? Anomalous result, the result that does not fit the pattern. So we have to search with each other which one of these data does not fit the pattern. Yes, if you concentrate well, we can find 170, which is anomalous, because this is far away from this 96 and 104. This is away, so this is anomalous result because does not fit the pattern. So what's meant by anomalous result? Result that does not fit the pattern. And then we have to draw a circle around the anomalous result, which is 170. Using the data for sample 3 in the table 2.2, calculate the percentage change in the vitamin C concentration of the apple juice when it is stored, and give your answer to 1 decimal place. Please concentrate about this part. The question here asks us to calculate the percentage change. Percentage change may be in a positive way and maybe in a negative way. Maybe this change increases, that this the final one, this more than the initial one, and maybe decreases. So it was more and becomes less. So when they said that the change, so we have to put the sign if it is in a negative side. So this means that the value decrease or the concentration decreases. So what are the data of sample 3? Return back. So we have the 272, this is the initial one, and 104, which is the final one. So we have the rule final minus the initial divided initial multiply 100. So 104 minus 272 divided 272 multiply 100. And by calculating this values, they give us negative 61.76. And they ask to give your answer to 1 decimal place. So 1 decimal place. So we have here this 1 decimal place, but after this value, this 6. So it will add 1 to the 7 to become 8. So this means that the percentage decreased by 61.8. So you have to put the negative sign. Next question, this figure 2.1 is a photograph of a fruit from a strawberry plant that has been cut in half. Draw a large diagram of a strawberry fruit as shown in the figure. So when we have this at any diagram, we have to make analysis for the diagram. What are the points that you have to know it? So we have here this part with the details. So we have many leaves, 1, 2, 3, 4, 5, 6 leaves, and we have some seeds. We have to put it in a consideration when we draw the diagram. And also we have to make it in a large in size and be sure that the outline of drawing should be not broken and be clear. To be easier more, I will learn you how can we draw it. Firstly, we can bring your pencil and make the outline of strawberry like this to make it easier to be printed in your mind. Okay, and then we have the stalk. After that, we have to draw different leaves. And one of them like this folded one. And another one this so folded. And so you have to make it by your pencil on the diagram to be easily printed in your mind. Then you start to draw. You just hold your pencil. This is preferable to be HB. This is the degree of graphite inside it. And then start to draw. My drawing, we can find it by this way. All the details that we can find it. We have the 6 folded leaves and we have many seeds and the shape with branches that inside strawberry plant. Line PQ in figure 2.1 represent the maximum diameter of strawberry fruit. Measure the length of line PQ. So the measurement of PQ may vary from each other and may be different from the mark scheme because it depends on the printed paper. So according to this size, so we can measure it. So no problem if your value is different from my value. So my value is PQ, which is 6.1, but we have to convert it into millimeters. So we have 6.1 centimeter, and 1 centimeter equals 10 millimeter. So by converting 6.1 multiply 10, so it will give us 61 millimeter. Calculate the actual diameter of the strawberry fruit using the formula. They give us the rule. We know that. We have also the magic triangle that make it easier. This magic triangle said, I am. I am. And then they asked about to calculate the actual diameter. The actual diameter, so which is equal image divided magnification. If you concentrate, well, we can find the magnification, which is equal 1.8 times. So 61 divided 1.8, which is equal 33.88. We ask about the three significant figure. So 1, 2, 3 significant figures. And after that, we can find the 8. So we can add 1 to the 8 to be 33.9. Next, the question describe a method that the student could use to test the strawberry fruit for reducing sugar. How can we test for reducing sugar? So we know that the Benedict solution used to test for reducing sugar, but we have here strawberry fruit. And we know that the strawberry, its color is red. So how can we test the strawberry fruit and we never see the changes that happens? So what we're going to do, they give us the strawberry fruit as it is. So we have to prepare a juice of strawberry and just make a filter using filter paper and then take the extract to make the experiment on it. So we never make this experiment on the direct strawberry fruit because it has a color. So we never see the color change easily. And we never know if it contains reducing sugar with a brick red color or this is the color of the original fruit. So we have to prepare a juice by mixing a strawberry with the water and mix them together and then filter this mixture and use the extract using Benedict solution. We have a condition for the Benedict test. So we have to apply heat using water bath at 80 degrees Celsius so we can find the change in color. If the color changes from blue, because this is the original color of Benedict solution, is blue, and if it changed into green or yellow or orange or brick red, so the fruit contains reducing sugar. So sometimes we can make this experiment directly, but in this experiment, we have to remove the red color of strawberry and just make it on the extract of the juice. Now we finished the exam, so I hope you enjoyed. Don't forget to like, share, and subscribe. We're waiting for the more and more exams. Thank you and goodbye.